right, welcome back. We've been looking at this new science of genetics. Would it surprise you to know that after looking at Gregor Mendel's work, as we did previously, that the inheritance of traits follows rules? And it's rules of probability. Probability is one of those things that you'd run across in a math class, and yet it finds its application here in biology. Remember, probability is a likelihood of an event occurring. And so the question here arises then at the beginning of our study of genetics is, to what extent can the inheritance of traits be predicted? All right, well, in order to study probability of events occurring in biology, we use a technique known as a Punnett Square, and it's a grid system that's used for predicting possible genotypes resulting from a cross of one individual to another. And so these axes that you see here, they represent the alleles that a parent would have. And so we'll put one parent on the top, we'll put one parent over on the left, and you can see the alleles here being represented by letters. It doesn't matter whether dad goes on the top or on the side. It really is gender neutral. You'll still get the same results. They'll just be sideways. And so we'll start off with the alleles of the parents, and then we'll fill in, we'll bring down those alleles that are inherited from parent one. We'll bring those down, and we'll bring those alleles that are inherited from this parent over here, we're calling parent two, and copy those across. And so we will get a pair of alleles that you see distributed through each of the boxes in the grid. And so that will give us a ratio of possible genotypes. Those genotypes represent phenotypes in the offspring. Well, just to review briefly, what we mean by genotypes are the actual pairs of alleles that we're representing by letters, as you can see here. The phenotypes that we're mentioning down here below are the actual traits that are expressed in the offspring and in the parent. Okay? Now, the simplest kind of Punnett Square that we can do, we looked at previously with Gregor Mendel's work, is a monohybrid cross, where we're involving just one trait at a time. And you can see we can examine the inheritance of a dominant, a homozygous dominant trait here up top against a homozygous recessive parent, as Gregor Mendel did. And you can see as we follow through and look at the genotypes that are produced by this cross, all of them, as Gregor Mendel observed, had purple flowers. Likewise, we can look at a heterozygous cross, as Gregor Mendel did in his F1 generation, and then we can examine the offspring of those F1 heterozygous purple-colored pea plants. And as you can see, the Punnett Square very nicely predicts out a 3-to-1 ratio of dominant to recessive that Gregor Mendel observed in his pea plants. Now, there's something also known as a ratio, a genotypic ratio, that we'll examine further. Here you can see 3 numbers that are separated by colons, and that's merely representing the homozygous dominant combination, FF, that you see in this first quadrant here, capital Fs. And then the second number are those that are heterozygous, capital F, lowercase f, which is in this second and then the third quadrant here. And then, of course, the last value here is going to be the last quadrant. In this case, it's lowercase f for both. And so we can see that we have a very nice prediction of what Gregor Mendel had observed in his experiments. And so this is wonderful because it now gives us a way of predicting, prior to doing an actual experiment, what the outcome is likely to be. I should say there's one qualification to that. That doesn't mean, for example, that if you had If you had 3 plants that grew up that were purple in this case, that the next one absolutely must have white petals on that. No, that's not the case. This is really a numbers game. And so we really have to look at several hundred individuals for these ratios to bear themselves out. And in fact, as you recall, that's what Gregor Mendel did. Okay? And so it's not going to predict necessarily what a particular individual would have. But it will, over the course of a number of individuals, you're going to begin to see this pattern expressed. And so we're getting back to that word probability. In this case, purple flowers are 3 times more likely, more probable to be expressed than the recessive white flower color. All right, and then we want to get into something known as a test cross. Let's say that you have a plant and you really don't know whether it's heterozygous or homozygous. This purple plant right here. And the only way that you'll really know is if you cross it with the recessive trait. In this case, this purple flower here on the left is heterozygous, then in the next generation, you're going to get a 1-to-1 ratio of the dominant to the recessive traits. That's what this would predict. Conversely, in this test cross, if this is a purebred purple flower, then the next generation, all of these will be purple. And so a test cross is used, as you can see, when you have an individual that is expressing the dominant phenotype and you don't know if its genotype is homozygous dominant or heterozygous. The best way is you cross it with a recessive individual, showing the recessive trait. Okay? Works every time. And then finally, a very important question that we want to try to answer here is, does the inheritance of one trait predict the inheritance of a second trait? For example, you may have observed that a lot of people with brown eyes also have darker hair. Well, is that always the case? Well, that's a similar kind of question that Gregor Mendel had when he was looking at the traits of the peas. And so what he decided to do was to do a cross between heterozygous peas across 2 traits at the same time. And this is what he discovered, that we would have 9 of those 16 that would express themselves, that would have the dominant trait of both characteristics. And then there would be a mixing of those traits where we have 3 that were dominant but showed the recessive trait in the second trait. And then the flip of that, the second trait was then dominant, but the first was recessive, but only 1 in 16 was recessive for both traits. And so this proved to Gregor Mendel something that we now know as independent assortment. And you remember when we studied meiosis, is that we learned that when gametes are produced by parents, that their combination of chromosomes are going to be randomly combined together when those gametes are formed. Well, Gregor Mendel could have predicted that outcome in his day as well, because this is what he observed in his pea plants, that the inheritance in this case of pea color, yellow here is dominant, it did not predict, it did not show that the smoothness and wrinkledness followed the exact same pattern. So these are actually inherited independently of each other. And that's why we're getting these 3s in the middle of the phenotypic ratio. And that's come to be called the law of independent assortment, as you can see, that states that allele pairs, they're going to separate independently during this process of meiosis. Now, I want to take a step back from that word law again. This is what you find in textbooks. And yet, as time has gone on, we found that there are important exceptions to that. And the exception is this: what if you have 2 traits that are inherited on the same chromosome? They're actually going to be inherited together then, aren't they? Unless crossing over occurs in between them, which we'll look at another time. But let's move on. Heredity patterns can be calculated with probability. As we said, probability is the likelihood that something will happen. And so we will want to look at the number of ways that a specific event can occur, dividing that by the total number of possible outcomes. And so this is how we calculate probability. And so it's going to apply to all kinds of random events, such as meiosis and fertilization. And we often model these kinds of processes using the flipping of coins. So when you flip a coin, you have heads or tails, don't you? So the probability of getting heads is basically 1 in 2, or 50%. And you can see as we look at the combinations possible, if you have 2 coins, then it's a 1 in 4 probability of getting both heads, a 2 in 4 probability of getting a head and a tail, because it can happen 2 different ways. And of course, there's only one way that you can get both tails, so that will be 1 in 4. Well, you can see that the Punnett Square comes in very handy in making these kinds of predictions for an inheritance. So in the classroom, we're going to apply these principles of probability using the Punnett Square. Great. I hope you took good notes. Thanks for watching. Bye-bye.